Hello everyone. Today both me and Ian are going to be talking about the similarities and differences in both the nucleotides DNA and RNA. To start off, the definition of DNA, deoxyribonucleic acid, is a nucleic acid that contains the genetic instructions used in the development and functioning of all modern living organisms. Its function now is to contain the biological and genetic instructions that control the day-to-day function of your cells. DNA structure is one that can be described as large double-stranded helix, which consists of two nucleotide strands containing a phosphate group, 5 carbons, and 4 nitrogen-containing nucleobases, which contain both purines and pyrimidines, adenine, thymine, cytosine, and guanine, A and G being purines, and T and C being pyrimidines.
Moving on to RNA, ribonucleic acid, which can be defined as a molecule that has various biological roles in coding, decoding, regulation, and expression of genes. Its function is to follow the blueprint of DNA and transfer the genetic code needed for proteins from the nucleus to the ribosome. Its structure is a smaller, single-stranded helix, similar to DNA, but RNA is composed of a phosphate group, 5 carbon sugars, and 4 nucleobases, one being different: adenine, uracil, guanine, and cytosine. A and G are both purines, and U and C being pyrimidines. Its location, depending on the specific type of RNA, can be located in the nucleus, the ribosome, or the cytoplasm.
Examples of RNA are messenger RNA, known as mRNA. This transcribes genetic information from the DNA found in a cell's nucleus, and then carries this information to the cell's cytoplasm and ribosome. Another is transfer RNA, known as tRNA. This is found in a cell's cytoplasm and is closely related to mRNA as its helper. tRNA literally transfers amino acids, the core components of proteins, to the mRNA in a ribosome.
Another form of RNA is rRNA, or ribosomal RNA, which is found in a cell's cytoplasm. In the ribosome, it takes mRNA and tRNA and translates the information they provide. From this information, it learns whether it should create or synthesize a polypeptide or protein.
According to Foreman Van Hemmert's journal on the nucleotide composition and structure of retroviral RNA genomes, retroviral RNA genomes have a biased nucleotide composition. The exact nucleotide usage pattern is distinct for different retroviruses. The nucleotide bias is even more in the single-stranded RNA domains. The exact nucleotide composition may provide the retroviral RNA genome with certain structural features. The similarities between DNA and RNA are that they both are polymers of nucleotides, they are both nucleic acids, and they are both macromolecules, meaning that they are created by polymerization of smaller subunits, known as monomers, and they are composed of thousands or more of atoms.
The differences between the two are that DNA uses deoxyribose sugar, has a helix, and excludes uracil as one of its nitrogenous bases, and in place has thymine. RNA uses a ribose sugar, is not helical, and excludes thymine as one of its nitrogenous bases, and in place has uracil.
So, knowing what we do now about both DNA and RNA, we can come to the conclusion that the differences far outweigh the similarities between the two, showing that they are two different molecules, but do work off of each other to make things flow and function, which is essential in all living organisms and their development, shaping us to be the unique and individual people that each and every one of us are.
This concludes our project on DNA versus RNA. Thanks for listening.